We've talked a lot in our course about post-translational modifications of enzymes and other proteins. And one of the most interesting parts of carbohydrate metabolism and the source of a lot of work in recent years has been the study of what's referred to as O-glycnacylation. O-glycnac is N-acetyl glucose linked by oxygens to groups on proteins, and O-glycnac signaling and the hexosamine pathway are discussed here. O-linked beta-N-acetylglucosamine, or O-glycnac, is a post-translational modification consisting of a single N-acetylglucosamine attached in O-beta glycosidic linkage to serines or threonine residues on proteins. Modification with a single O-glycnac is a signaling device similar to phosphorylation. O-glycnac signaling and its crosstalk with phosphorylation modulate a wide variety of cellular events. And signaling and crosstalk like this turn out to be altered in diseases like diabetes and cancer. O-glycnacylation plays a direct role in insulin signaling and cancer. And that pathway is shown here, and this is really part of what's called the hexosamine pathway, and we should take it in parts. Up at the top of the figure, you see glucose metabolism, which of course involves glucose being converted in glycolysis, and amino acid metabolism, which we haven't covered yet, but which can produce free amino acids like glutamine. And then fatty acid metabolism, shown here, produces acetyl-CoA. Acetyl-CoA is the primary breakdown product of fatty acids. And when we get to chapter 26 and look at nucleotide metabolism, we'll see that that's where nucleotides like UTP come from. So you can put all this together in what's called the hexosamine pathway, and we'll take a look. Glucose can be converted, of course, to glucose 6-phosphate and then fructose 6-phosphate, and then an enzyme referred to as the glutamine fructose 6-phosphate aminotransferase takes glutamine and fructose 6-phosphate as substrates and produces glucosamine, a glucosamine 6-phosphate in fact. You can also produce glucosamine 6-phosphate directly from glutamine without involving fructose 6-phosphate, and so either way you end up with glucosamine 6-phosphate as shown here. And that can be acetylated by acetyl-CoA to produce an N-acetylglucosamine 6-phosphate, what we refer to as glycnac. And that can be converted to N-acetylglucosamine 1-phosphate, which can combine with UTP to produce UDP-glycnac. That's a sugar nucleotide with an N-acetyl group on a glucose. And that turns out to be an important signaling device. O-glycnac transferases take proteins with serines and threonine residues and add the O-glycnac to them, and that can activate or inhibit processes all through the body and all through cells. And notice that the arrows here indicate that O-glycnacylation is increased in diabetes and decreased in cancer. There's a few other aspects of this that we'll consider in later chapters, but it's important to appreciate that all these different metabolic pathways are involved in producing the sugar nucleotide that is N-acetylglucose linked to a UDP glycnac. We'll talk more about this as we go along. The last topic in this chapter involves the production of electrons for biosynthesis from glucose, and this is something referred to as the pentose phosphate pathway. Cells are provided with a constant supply of NADPH for biosynthesis by the pentose phosphate pathway. This is also called the hexose monophosphate shunt, and you may see it described in either way. This pathway also produces ribose 5-phosphate, and the pathway consists of 2 oxidative processes followed by 5 non-oxidative steps. It operates mostly in the cytosol of liver and adipose tissue. Adipose tissue is fat cells, essentially. NADPH is used in the cytosol for fatty acid synthesis, as we'll see in chapter 24. The whole pathway is shown here, and it's complex, so we'll take it in parts. This is the pentose phosphate pathway. The numbers in the blue circles indicate the steps that are discussed in your text, and we'll take a look at these one by one. First, here are the oxidative steps that we mentioned. A glucose 6-phosphate is converted to a 6-phosphogluconate, and then to a ribulose 5-phosphate. Both of these are oxidative processes. Here the aldehyde is converted to an acid, and in the next step, another oxidation occurs with release of CO2. In both cases, the coenzyme required for the process is NADP+, and NADPH is the result. And NADPH electrons can be used for reductive anabolic processes. This is where most of the electrons for anabolic biosynthesis comes from. And notice over here that steps 4 and 5 go on, as shown in the next slide. And here are those next steps. This is the ribulose 5-phosphate from the previous slide. It can either react to produce ribose 5-phosphate or xylulose 5-phosphate. And we'll come back to xylulose 5-phosphate, which turns out to recently have been discovered as a metabolic regulator. The ribose 5-phosphate that's produced here can be used, of course, for nucleic acid synthesis to make DNA and RNA. This is really the source of the ribose used to make DNA and RNA. Step 6 is an interconversion that involves ribose 5-phosphate and xylulose 5-phosphate to produce cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. If you think about it, xylulose 5-phosphate is a 5-carbon metabolite, and so is ribose, so that's 10 carbons altogether. And these are exchanged to give you a 7-carbon sugar and a 3-carbon sugar. Reaction 7 takes the 7 and the 3 and makes a 4 and a 6 out of it. You get an erythrose 4-phosphate and a fructose 6-phosphate. And then, in step 8, another xylulose 5-phosphate and an erythrose 4-phosphate, that's a 5 and a 4, can make a 6 and a 3, the fructose 6-phosphate and a glyceraldehyde 3-phosphate shown here. So all these steps interconvert small sugars one to another, and all of these can be used in various ways in metabolism. Let's take a look at the reactions one by one. First, the oxidative steps of the pentose phosphate pathway. The first reaction is the glucose 6-phosphate dehydrogenase. It's an irreversible first step in a pathway, so it's highly regulated. And you can imagine it has a large negative delta G. The next reaction is the gluconolactanase, and it's a reaction that really isn't needed because the uncatalyzed reaction happens too in cells. But the second oxidation is really an oxidative decarboxylation, and we've seen these before several times in the pyruvate dehydrogenase complex and the TCA cycle, and it's called the 6-phosphogluconate dehydrogenase. And as noted, it's an oxidative decarboxylation in that order. Oxidation occurs first, followed by decarboxylation. Here's the glucose 6-phosphate dehydrogenase reaction, which turns alpha-D-glucose 6-phosphate into 6-phospho-D-gluconolactone. NADP+, of course, is the nicotinamide that's used for this, and NADPH electrons are the result. Here's the gluconolactanase reaction that turns the 6-phospho-D-gluconolactone into a 6-phospho-D-gluconate. This reaction also occurs in the absence of an enzyme. And this shows you the 6-phosphogluconate dehydrogenase reaction, which takes 6-phospho-D-gluconate and turns it into ribulose 5-phosphate, with an intermediate shown that is a 3-keto 6-phospho-D-gluconate. In this reaction, the initial NADP+ dependent dehydrogenation yields a beta-keto acid, 3-keto 6-phosphogluconate, which is very susceptible to decarboxylation in the second step. The resulting product, D-ribulose 5-phosphate, is the substrate for all the non-oxidative reactions of the pentose phosphate pathway. Let's take a look at the non-oxidative steps. There are 5 steps, but really only 4 kinds of reaction. The phosphopentose isomerase reaction converts a ketose to an aldose. The phosphopentose epimerase does an epimerization at carbon 3. The transketolase reaction is thiamine pyrophosphate dependent and transfers 2 carbon units. And finally, the transaldolase reaction uses a shift base mechanism to transfer 3 carbon units. The phosphopentose isomerase reaction converts, as we mentioned, a ketose to an aldose with an enediol intermediate, as shown here. The phosphopentose epimerase reaction interconverts ribulose 5-phosphate and xylulose 5-phosphate. This mechanism also involves an enediol intermediate and occurs with inversion at carbon 3. The transketolase reaction of step 6 in the pentose phosphate pathway is shown here. Xylulose 5-phosphate and ribose 5-phosphate are converted to glyceraldehyde 3-phosphate and cetoheptulose 7-phosphate. This is a 2-carbon transfer that requires thiamine pyrophosphate as a coenzyme. We discussed thiamine chemistry back in chapter 19, and you should look at that in the pyruvate dehydrogenase reaction. The transketolase reaction in step 8 is shown here. This is another 2-carbon transfer, and it also requires thiamine as a coenzyme. It interconverts xylulose 5-phosphate and erythrose 4-phosphate with glyceraldehyde 3-phosphate and fructose 6-phosphate. The mechanism of the thiamine pyrophosphate-dependent transketolase reaction is shown here. The group transferred is really an aldol, and despite this, the name transketolase persists for this enzyme. So you see here the thiamine anion that is formed attacks the carbonyl carbon. The second step involves proton removal followed by a carbonyl formation to make the glyceraldehyde 3-phosphate product as shown here. And the thiamine 2-carbon unit goes on in the next step to form this resonance stabilized anion, which can then attack the carbonyl carbon of ribose 5-phosphate, producing this intermediate, followed by proton removal to form the cetoheptulose 7-phosphate, and the thiamine pyrophosphate is regenerated as shown here. The transaldolase reaction involves transfer of a 3-carbon unit, shown here, from cetoheptulose 7-phosphate to glyceraldehyde 3-phosphate to produce erythrose 4-phosphate and fructose 6-phosphate. The transaldolase mechanism involves a Schiff base, and that's shown here. Here's the lysine amino group attacking the carbonyl carbon to form the Schiff base, and just as we saw in the aldolase reaction in glycolysis, proton removal here leads to cleavage to produce the erythrose 4-phosphate product, and this resonance-stabilized enamine, which can break down by attack on glyceraldehyde 3-phosphate to form this adduct, and then water cleaves the Schiff base to yield the fructose 6-phosphate product. You should be familiar with this mechanism. The utilization of glucose 6-phosphate in the pentose phosphate pathway depends on the cell's needs for ATP and NADPH and ribose 5-phosphate. Glucose can be a substrate either for glycolysis or for the pentose phosphate pathway. The choice really depends on the relative needs of the cell for biosynthesis and for energy from metabolism. Glycolysis, of course, provides energy from metabolism, whereas the pentose phosphate pathway provides the biosynthetic NADPH and ribose 5-phosphate. ATP can be made if glucose 6-phosphate is sent to glycolysis, or if NADPH and ribose 5-phosphate are needed for biosynthesis, glucose 6-phosphate can be directed to the pentose phosphate pathway. Depending on these relative needs, the reactions of glycolysis and the pentose phosphate pathway can be combined in 4 principal ways, as we'll describe here. First, if both ribose 5-phosphate and NADPH are needed by the cell, the first 4 reactions of the pentose phosphate pathway predominate. NADPH is produced and ribose 5-phosphate is the principal product of carbon metabolism. On the other hand, if more ribose 5-phosphate than NADPH is needed by the cell, then synthesis of ribose 5-phosphate can be accomplished without making NADPH by bypassing the oxidative reactions of the pentose phosphate pathway. Here's case 1. If both ribose 5-phosphate and NADPH are needed, the first 4 reactions of the pentose phosphate pathway will predominate, and the principal products, as shown here, are ribose 5-phosphate and NADPH. Case 3 is if more NADPH than ribose 5-phosphate is needed by the cell. This can be accomplished if ribose 5-phosphate produced in the pentose phosphate pathway is redirected to produce glycolytic intermediates. And in case 4, we imagine that both NADPH and ATP are needed by the cell, but ribose 5-phosphate is not. This can be done by redirecting ribose 5-phosphate, as in case 3 above. The fructose 6-phosphate and glyceraldehyde 3-phosphate made in this way can then proceed through glycolysis to produce ATP and pyruvate continues through the TCA cycle to make more ATP. There's an interesting description in your book about the integration of the Warburg effect. ATP consumption promotes cancer metabolism, the original postulate by Warburg. Cancer cells use glycolytic intermediates for the synthesis of macromolecules, and they must balance their ATP requirements and their biosynthetic needs. Xiaodong Wang has shown that NTPD5, which is a UDPase in the endoplasmic reticulum, is associated with ATP consumption in tumor cells. UMP made in this reaction is transported into the cytosol, where it's converted back to UDP and then UTP, and then to UDP glucose. Ensuing reactions send glycolytic intermediates to the pentose phosphate pathway, helping cancer cells to fine-tune production of NADPH, ribose 5-phosphate, and ATP. Let's have a look. All of that is shown here. The NPD5 is located right here, and in this pathway, glucose can stimulate sugar nucleotide synthesis to make UDP glucose, which enters through a transporter into the endoplasmic reticulum, and UDP glucose is used in the ER to produce glycoproteins, as shown here. The sugars of glycoproteins are provided by the UDP glucose, the activated form of glucose that is a sugar nucleotide. And in the reactions that do this, UDP is released, and the UDP is converted by the NPD5 UDPase to make UMP, which exit the ER, and that UMP goes through this process here to convert ATP and UMP to UDP and ADP. And that ADP can go through the adenylate kinase reaction, which produces both ATP and AMP, and the AMP produced here will lower the ATP to AMP ratio, which stimulates the phosphofructokinase and therefore stimulates glycolysis. And now we'll look at cellulose 5-phosphate, which has recently been shown to be a metabolic regulator. In addition to its role in the pentose phosphate pathway, cellulose 5-phosphate is also a signaling molecule. When blood glucose levels rise, glycolysis and the pentose phosphate pathways are activated in the liver, and the latter pathway makes cellulose 5-phosphate, as we've seen. And this turns out to stimulate protein phosphatase 2A, also known as PP2A. And this phosphatase dephosphorylates the interesting bifunctional enzyme that we looked at earlier in this chapter, the PFK2 fructose 2,6-bisphosphatase enzyme. And PP2A also dephosphorylates the carbohydrate-responsive element binding protein, ChREBP. The result is that glycolysis and lipid biosynthesis are both activated, as we'll see. And that's shown here. Cellulose 5-phosphate stimulates the protein phosphatase 2A. And if we look down here, you'll see that the ChREBP is phosphorylated normally, but can be dephosphorylated by the protein phosphatase 2A. And then, of course, it can be rephosphorylated by protein kinase A, as shown here. Stimulation of the protein phosphatase 2A also stimulates the phosphofructokinase 2 activity of the bifunctional enzyme and inhibits its fructose 2,6-bisphosphatase activity. And both of these effects increase the levels of fructose 2,6-bisphosphate in the cell. You'll remember that fructose 2,6-bisphosphate stimulates glycolysis, which produces pyruvate, and then in the pyruvate dehydrogenase reaction you produce acetyl-CoA, which can provoke and stimulate lipid synthesis. Lipid synthesis is also stimulated by the dephosphorylated form of the ChREBP. So in the end, the cellulose 5-phosphate that we mentioned as a regulator carries out both of these processes by activation of the protein phosphatase 2A. The last section in Chapter 22 discusses the aldose reductase reaction and diabetic cataract formation. Diabetics suffer many consequences of their disease, and one is the propensity for cataract formation in later life. All type 2 diabetics are encouraged to have eye exams on a regular basis, and one of the things that's looked for in these exams is cataract formation. It turns out that hyperglycemia is the cause. But why should high sugar levels cause cataract formation? Evidence points to the so-called polyol pathway, in which glucose and other simple sugars are reduced in NADPH-dependent reactions. Glucose is reduced by the aldose reductase to sorbitol, which accumulates in the lens fiber cells in the eye, increasing pressure and eventually rupturing the cells, and that's the cause of cataracts. Aldose reductase inhibitors such as tolrestat and epalrestat suppress cataract formation, and these are shown here. This is the aldose reductase reaction on the left that converts glucose to sorbitol, And here are the structures of tulrestat and epulrestat. You certainly don't need to memorize those structures. But as noted here, glucose is reduced by aldose reductase to sorbitol, which accumulates in lens fiber cells, increasing pressure and eventually rupturing the cells and causing cataracts. Tulrestat and epulrestat suppress this cataract formation. And that concludes Chapter 22. We'll see you in Chapter 23.